All right. Hi, everyone. I hope you're having a good evening. Welcome again to our another workshop. Again, I apologize for the discrepancy that happened yesterday, but today, get excited for a new workshop. Today, we have Emma presenting on virology. And thank you again, Emma, for coming to present. I saw the slides and they're so amazing. So whenever you're ready, you can share your slides and begin.
Okay, awesome. I will share my screen. Thank you guys for coming. Okay, share. Okay, did it work? It's saying it needs permission.
Oh, that's weird. It says all can. Okay, let me. Oh. I think I allowed permission. Maybe
Okay, I'll try one more time.
again. Yeah. Okay.
Okay, cool.
Yeah, now it worked.
Awesome. All right, so I'm going to be going over the basics of viruses today. Seems pretty relevant right now in today's society. But yeah, I'll start. So what is a virus? A single virus particle is known as a virion. It is an infectious agent made up of nucleic acid, either DNA or RNA, and I'll go into the different types later on, surrounded by a protective protein coat or shell called a capsid. Certain virus strains will have an extra layer surrounding it, a lipid bilayer called an envelope. So not all viruses have an envelope. There's a picture of it. Are viruses alive or dead? Simply put, viruses don't count as living organisms as they don't have their own metabolism and they can't generate their own energy. They have to utilize a host cell's energy. So there's kind of a living organisms checklist that scientists go through to determine this. Cells can maintain homeostasis. They have their own nucleus, organelles, and cytoplasm, and viruses have none of these things. They cannot reproduce, but they can replicate within a host cell. They cannot create their own energy. Again, they don't have their own metabolism, but they can utilize the energy of a host cell in their energy. They cannot respond to stimuli. There's no evidence of that yet. They can evolve and adapt to their environment through mutations. So that's why there's different strains, and that's why it's not necessarily considered dead, but it is not considered a living organism. So just some basic terminology. A reservoir of an infectious agent is the habitat in which the agent normally lives, grows, and multiplies. So reservoirs can be humans, animals, or the environment. The reservoir may or may not be the source from which an agent is transferred to the host. For example, the reservoir of Clostridium botulinum is soil, but the source of most botulism infections is improperly canned food containing the spores of that virus. Or bacteria. Many common infectious diseases have human reservoirs, diseases that are transmitted from person to person without intermediaries, including sexually transmitted diseases, measles, mumps, strep, and many other respiratory pathogens, which I'll go into later as well. Because humans were the only reservoir for the smallpox virus, naturally occurring smallpox was eradicated after the last human case was identified through vaccines. A host, again, is a large organism, any organism that harbors a virus in some sort of symbiotic relationship. All viruses depend on the host in order to reproduce within it. Again, viruses cannot replicate on their own. And then a vector is an agent that carries and transmits an infectious pathogen into another living organism, or a DNA molecule used as a vehicle to artificially carry foreign genetic material into another cell, so genetically modified organisms. Transmission is the process in which a virus is spread between hosts, hosts being animals, bacteria, plants, anything that it can live in and infect. So different types of transmission. There's airborne, small airborne particles less than 5 millimeters, or dust particles within infectious agents. Dispersed by air currents, or they can stay in the air for long periods of time. Example of this is tuberculosis. There's droplets, coughing, sneezing, talking. Example of this is influenza. There's contact, like the norovirus, so direct body surface to body surface contact. And then there's indirect, between person and contaminated object. And then that vector would be called a fomite. A common vehicle for this is contaminated food, contaminated water, medications, or medical devices. So a needle transmitting HIV. Latency is a virus's ability to lay dormant, inactive, and then be reactivated later to become infectious, like the herpes zoster. This is different than opportunistic bacteria, but it's a very similar process. And then tropism, which I'll go into later, is specific tissue or cells that some viruses infect within the host. So the rabies virus actually targets your nervous system tissue and can shut that down pretty quickly. So it's actually pretty bad virus to get once you're infected with it, and it can cause you to be paralyzed. So it infects certain cells and tissues within people. That's tropism. Let's see. Okay, so different classifications of viruses. You can classify it based on the genome properties, so RNA versus DNA, and then the different types. There's something called Baltimore classification, which I won't really go into, but it's the types of mRNA synthesis that the virus undergoes. Morphology, so size and shape, presence of an envelope or no envelope, the structural proteins, which also helps determine its molecular weight, and the targeted host, so whether it targets animals, plants, bacteria, or more than one. Going further into virus classification by genome, viruses have either DNA or RNA genomes. It can be linear or circular, single-stranded, positive, negative sense, or double-stranded. And then all the different DNA and RNA molecules within viruses vary in length. And there's a couple examples at the bottom of which ones are which. So DNA, linear, parvovirus, herpes virus, M13, baculovirus, TMV, reovirus, hepatitis D. Target host classification. So bacteriophages are viruses that infect bacteria. So when an infection of a cell by a bacteriophage results in the production of new virions, the infection is said to be productive. If the virions are released by bursting the host cell, the virus replicates by means of a lytic cycle. So there's lytic and lysogenic cycles when they're infecting the different hosts. An example of a lytic cycle bacteriophage is T4, which infects E. coli found in the human intestinal tract. Sometimes, however, a virus can remain within the cell without being released. For example, when a temperate bacteriophage infects a bacterial cell, it replicates by the lysogenic cycle. And the viral genome is incorporated into the genome of the host cell. When the phage DNA is incorporated into the host cell genome, it is called a prophage. So an example of a lysogenic bacteria would be the lambda virus, also infects the E. coli bacterium. Viruses that infect plant or animal cells can also undergo infections where they are not producing virions for long periods. An example is the animal herpes virus, including herpes simplex virus, the cause of oral and genital herpes in humans. In a process called latency, these viruses can exist in nervous tissue for long periods of time without producing new virions, only to leave latency periodically and cause new lesions in the skin where the virus replicates. So you've heard of the herpes virus being latent or active. And even though there are similarities between lysogeny and latency, the term lysogenic cycle usually reserved to describe bacteriophages. So those are the three types of target host classifications. Going a little bit further into animal viruses, unlike viruses of plants and bacteria, don't have to penetrate a cell wall to gain access to the host cell. So non-enveloped or naked animal viruses, so viruses without an envelope, which I'll go into later as well, can enter cells in two different ways. As a protein in the viral capsid binds to its receptor on the host cell that it's targeting, the virus can be taken inside the cell via a vesicle during the normal cell process of endocytosis. An alternative method of cell penetration with animal viruses, viruses that are targeting animal hosts, is used by non-envelope viruses is for capsid proteins to undergo a shape change after it binds to the receptor on the host cell. This creates a channel in the host cell membrane, allowing its genome to be injected into the host cell through the channel. So it can just strictly inject its RNA or DNA into the host cell to be replicated. All right, let's see. After they make the new proteins and copy their genomes within the animal host cell, animal viruses complete the assembly of their new virions and exit the cell. This can occur by lysis or apoptosis, where the host cell essentially dies. see. So again, some viruses have an envelope, some do not. Enveloped viruses, viruses containing only the genome, RNA and DNA, and the protein capsid are considered non-enveloped viruses, while some viruses have another membrane surrounding the capsid, as we've discussed, that contain proteins and lipids, and that's considered the envelope. So classification of viruses by morphology and shape. There's helical, icosahedral, complex virus, and spherical, and there's just some examples of what they look like. How does the virus spread? It spreads by replication within the host cell. Again, it never divides on its own, and it requires the host cell and the host cell's energy and metabolism. So just basic overview of the steps, and there's a picture to go along with it. Step 1 is attachment to the host cell at the receptor, which I'll go into in the next slide. Step 2 is penetration or entry into the host cell, which I've discussed can be endocytosis, fusion, or translocation. So either completely going within the host cell in a vesicle as one, and then bursting within the host cell by lysis, or just injecting its RNA or DNA into the host cell. There's uncoding, so released genome travels to the transcription translation sites of the host cell. Then there's genome replication, assembly, which is formation of the new virion, maturation, and release. Going further into attachment, so viruses are host specific and enter into the host or target cell that is trying to target. Again, it could be bacteria, it could be plant, it could be animal hosts. No cellular or metabolic energy is required for the attachment phase. Cellular tropism, as mentioned, is certain kind of viruses attacking specific types of tissues or cells, like the rabies virus attacking your nervous tissue. Again, examples of the virus and the cell types that it attacks: HIV, T macrophages and lymphocytes, T lymphocytes and macrophages, rabies attacking your muscle and neurons, hepatitis A, B, and C attacking liver, which are hepatocytes. And viruses can have narrow or broad host range. So rabies is a pretty broad range virus. It can infect many different mammals, while HIV is a pretty narrow range virus. It mainly infects humans and or chimpanzees. And then going a little further into attachment, viruses, there's a picture of it. Viruses use host cell receptors and anti-receptors on the capsid and outer envelope to attach to the host cell. So on the capsid or the envelope, you're going to have proteins or glycoproteins that are embedded, and those will attach to different receptors on the target host cell. An example of this is HIV attaches to CD4 receptors. The rabies virus attaches to acetylcholine phospholipids. How vaccines essentially work. The body is exposed to a weakened or dead virus. The body's immune cells make antibodies to attack the pathogen. If encountered a second time, the body is prepared to fight the virus with antibodies. So when an individual is exposed to pathogens that causes the infectious disease through natural infection, the body's immune system responds by having its immune cells, like white blood cells, replicate and target these pathogens with antibodies. Antibodies are blood proteins that bind to pathogens and essentially destroy them. Once the pathogen has been destroyed, the immune cells disband and immune system activity returns to normal levels, but key cells known as memory cells remain. If the individual is exposed to the same virus in the future, these memory cells can identify the pathogen and respond incredibly quickly with the appropriate antibodies. So different types of vaccines. There's the live attenuated vaccine, which contains a weakened pathogen and usually requires 1 to 2 doses, and there are some examples. Again, these vaccines create a strong and lasting immune response. Just 1 or 2 doses of most live vaccines can provide a lifetime of protection. Examples: measles and mumps, rubella, the MMR, and the rotavirus and varicella vaccines. Inactivated vaccines use a killed form of the pathogen. These vaccines typically don't provide protection that is as strong as live attenuated vaccines, so several doses are usually administered over time to elicit that ongoing immunity. These additional doses are called booster shots. Examples of these vaccines are hepatitis A, influenza, and rabies vaccines. And something to note about rabies vaccines, it's super expensive and limited. So people usually only get the rabies vaccine if they are prone to rabies. So vets, farmers, people that work out in wildlife, people that are more likely to be exposed to rabies, or if someone has been known to have been bitten by an animal that could potentially have had rabies. So it's very rare, but it's very important to get the rabies vaccine as soon as you feel you may have been bit or exposed, or you are prone to being around an infected agent. Subunit vaccines, like inactivated vaccines, do not contain live pathogens, but they are distinct in that they contain only select antigenic parts of a pathogen. So the parts that are necessary to elicit a protective immune response. These also require booster shots, and some examples of subunit vaccines are HPV and pneumococcal vaccines. Lastly, toxoid vaccines use a toxin or a harmful product made by the pathogen that causes the disease. This means the immune response is targeted to the toxin instead of the whole pathogen. These also may require booster shots. Examples of these are diphtheria and tetanus vaccines, as well as, okay. So viral mutations in different strains and essentially like creating the vaccine. This is something that I learned in a virology class recently, which is pretty cool. And I didn't know this before. So the flu shot is different every year due to mutations and reassortment, which I'm not going to go into detail in this, but there's some really good videos on it that explain the different ways that, for example, the flu vaccine undergoes reassortment to create different strains, whereas the measles, mumps, and rubella are not segmented. So the flu season in this part of the world, the United States, I'm not sure where a lot of you are from, but is around August or early September to early May. And then on the other side of the world in Australia, it's the opposite season. So they have completely opposite flu seasons. We base our flu shot for an upcoming year based on what the flu virus was like in Australia in their flu season. So we essentially make it in the summer based on their strain. We assume that that's the strain that we'll get. Then based on the strain that we experience over here, they base their vaccine for that next year on that, which is kind of cool. And obviously there's a lot of room for uncertainty. There's no way to know exactly how it will replicate or cross over or end up over here. So essentially scientists make their best bet. Does not mean you shouldn't get the flu shot, even though it's not 100% effective, because as we know, it helps mitigate symptoms. And then lastly, I wanted to wrap up the presentation with the fun fact. The first virus discovered is the tobacco mosaic virus, TMV, in 1892. It was deemed a virus in 1930. It is a single-stranded RNA virus. That's what it looks like on the right. It infects a wide range of plants, so it's considered broad-range virus. It's caused distinct patterns, mosaic-like patterns, and discoloration on leaves, as you can see in the picture. And Wendell Stanley crystallized sap from sick tobacco plants to discover its makeup. Thank you. That is the presentation.
Thank you so much for that very informative presentation, Emma. I definitely learned a lot about virology and viruses, and as I'm sure everyone else did. Does anyone have any questions for either of us or anything like that? If not, I will put the attendance form in there, and please feel free to ask your questions.
Perfect.
So the attendance form is going to be due at 7 p.m. Eastern tomorrow. So I've left it open for a little bit.
If anyone is interested, I can also send over the virology course that I took was pretty expensive. So I graduated and I work in research now. I just graduated this past year. So I know I would not have been able to have afforded that class in undergrad. If anyone wants me to send over the slides, it's super interesting and I can send that to you, email that to you. It was 5 classes. So it's like 5 PowerPoints, but just let me know. Here, I will put my email in the chat and you can email me if you want the slides from the virology course. Some of it is on the MCAT, so might help. Yeah, I do research in neuroscience right now. So we look at brain tsunamis after an injury has occurred and kind of what happens to the brain and the memory and everything. It's cool. It's definitely look it up. If you type in, there's not a ton of research on it right now. So if you type in brain tsunamis and research, you could type in University of Cincinnati as well. And some cool research will come up.